So today we are going to discuss Ames test. Okay, so for Ames test, we take a test tube and we add the tissue called as the rat liver extract. Okay, along with that we add the bacteria, Salmonella bacteria. Okay, so the function of this tissue is that this tissue provides the necessary nutrients for the growth and the survival of the bacteria. This tissue provides the necessary nutrients for the growth and the survival of this bacteria, Salmonella. Now this bacteria that we are choosing, it has a characteristics, that is this bacteria Salmonella that we are choosing this bacteria can synthesize almost all the nutrients, but it will not be able to synthesize the amino acid histidine. Okay, so such bacteria which can synthesize almost all the nutrients that it is required for its normal growth and proliferation, but it will not be able to synthesize one particular nutrient. We call those types of bacteria, or more specifically those type of mutants, as auxotrophic mutants. Okay, so what are auxotrophic mutants? Auxotrophic mutants are those mutants which will be able to synthesize almost all the nutrients that will be required for the growth and proliferation, but will not be able to synthesize one particular nutrient. Okay, they will be deficient in synthesizing one particular nutrient. Okay, so here we are choosing the histidine, this amino acid histidine. So this bacteria will be lacking or will not be able to synthesize amino acid histidine. Now why we are using this amino acid histidine? Because this amino acid, we will later, you will see this amino acid will be used as some sort of a differentiator. Okay, so by this amino acid we will differentiate between the normal strains of bacteria and the mutagenic strains of bacteria. So what we are doing now, we are taking this test tube where we have added rat liver extract and Salmonella bacteria, and we are doing two processes. First, we are part of the contents is plated on a medium with minimal histidine. Okay, so minimal histidine means it almost contains no histidine or very small amount of histidine. So part of the content, okay, rat liver extract plus Salmonella strain, part of it is added to a media with containing minimal histidine. So this media that we are adding here, this is used as a control. Okay, so this is the first process. So next process, another process what we are doing after adding part of it, another part of it, we are mixing the contents of this test tube with another test tube. Okay, we are mixing the contents of this test tube with another test tube that is suspected to contain a possible mutagen. That means this test tube is suspected to contain a possible mutagen, but we are not yet confirmed. So let's see. So part of this rat liver extract plus Salmonella strain, part of this test tube is mixed with the contents of this test tube containing possible mutagen. Okay, so we are mixing it into a third test tube, contents of this test tube, and we are plating it into another medium. Okay, plating it. So this is the second procedure. We are plating it into another medium. This medium also contains minimal histidine. So what is the motive of using a media containing minimal histidine is that, so those Salmonella strains which requires histidine for the growth, they will not be able to grow in a medium containing minimal histidine because histidine is almost not present. And for these kinds of Salmonella strain, histidine is very much essential for the growth. Otherwise, without histidine, they will not be able to grow. Okay, so we will see which strains of bacteria will be able to grow in this medium and which strains will not. Okay, so in the control step, as you can see, after incubation, where almost all the bacteria dies. Okay, almost no colony, only 2 colonies. So these are natural revertants. Revertants means they were previously not mutagenic, but somehow they have become mutagenic. So except 1 or 2, almost all the bacteria have died. That means these bacteria were the natural bacteria which couldn't survive in this medium. But after the addition of this possible mutagen in this second phase of the experiment, you will see almost all of the bacteria have grown in this minimal medium. Okay, so minimal medium containing histidine. That means these bacteria have reversed. That means this bacteria have become mutagenic, because why, previously Salmonella strain couldn't synthesize histidine. So ideally they would not have grown in this minimal medium. but now they are growing in this minimal medium containing histidine. They are forming colonies. So what does that mean? They do not require histidine anymore for their growth. That means they can synthesize histidine within their body. So they do not require addition of histidine from outside. That means they have become mutagenic. So this is depicted here. High number of revertants from histidine minus. Histidine minus means they previously could not able to synthesize histidine and they require addition of histidine from outside. And now they have changed to histidine plus. That means they do not require any further addition of histidine from outside. They can synthesize within their body. So such as mutagen causes mutations. Now what happens if I explain here? What happens? These auxotrophic salmonella strains, they had an operon. Okay, suppose this is the operon, and here is the gene for histidine synthesis. And previously what happened, this gene could not synthesize histidine. Okay, so that's why they require addition of histidine from outside. But when you add the possible mutagen, probably this mutagen caused some sort of frame shift mutation. So suppose this is the operon again, and here you have the gene for histidine, and this, when you are adding this mutagen from outside, possible mutagen from outside, it is causing a sort of frame shift mutation, and because of that now this operon can synthesize histidine. Okay, so now this operon can synthesize histidine. So now these bacteria do not require any further addition of histidine from outside. So even if the media doesn't contain histidine, because it is minimal histidine media, as you can see, so the bacteria are still growing in this media. They can still grow in the media because now they can synthesize histidine within their body. Okay, this mutagen is causing some sort of frame shift mutation. As a result, what is happening? Histidine can be synthesized within their body now, and these bacteria now can grow within the media containing minimal histidine. So they do not require any further histidine to be added from outside for the growth. They can now synthesize histidine and they can easily grow. That is why you can see in the control where you have not added the mutagen, almost all the bacteria have died because they couldn't synthesize histidine, and you have not added histidine on the media also. So they almost died. Almost all the bacteria died. But in this experiment where you have added the mutagen, you can see almost all the bacteria survived because this mutagen caused a frame shift mutation, and as a result these bacteria now can synthesize histidine and they do not require any further addition of histidine, or they do not require any further supplementation of histidine, okay, for their growth. They can now synthesize it within their body, and even if you don't add it to the media, it won't be a problem because now they can grow.